So let us see the development of the definitive heart formation. For that, we have to go in sequence from how the heart tube is being formed, and then the chambers, then the septations, and so on. So with the development of the head fold of the embryo, the two paramedian endothelial tubes of the heart lie ventral to the foregut, and they project into the dorsal wall of the pericardial sac, which is covered by myoepicardial mantle. So after the head fold has happened, this yellow one is the foregut, this red one is the primitive heart tube that has been formed. So the heart tube is ventral to the foregut. Ventral to the heart tube is the pericardial sac, into which the developing heart will bend and enter into it. So this is where is the folding of the embryo hasn't yet occurred. So this is the sagittal section. If you come from dorsal to ventral, these are the structures you find. This is the pericardial sac, then it is the myoepicardial mantle, then it is the cardiac jelly, then it is the primitive heart tube that has fused to form a single heart tube. So these are the two paramedian endothelial tubes which are you are seeing in front of the pericardial sac here, the white thing. So they fuse from craniocaudally to form a single heart tube. So with the cephalic and the lateral foldings, the embryo will bring the two lateral foldings, the lateral endothelial tubes, closer in the thoracic region, where they fuse to form a single tube, and the fusion occurs in craniocaudal direction. And here, before the cephalic fold, the cranial end is the venous end and the caudal end is an arterial end. So once the cephalic folding occurs, there is reversal of these ends also, where now the cranial end becomes the arterial end and the caudal end of the developing heart tube becomes the venous end. And the timeline goes like on day 21, the single heart tube is being formed, and as soon as the single heart tube forms, the heartbeat is developed. So heart starts its beating on day 22, and on day 23 heart begins to loop into the pericardial sac, and on day 24 blood starts circulating, and the circulating blood is very, very important for the further development of the heart tube. And by day 28 the looping is complete. So the cranial end of a single tube may be called as an arterial end of the heart, and it bifurcates into two branches, that is the aortic arches. This is the first aortic arch which joins with the dorsal aorta, encircling the developing foregut in the cranial end. And if you can see here, this is the one. So from the cranial end, the arterial end, the single tube may be called as a first aortic arch, which flank the developing foregut and join with the dorsal aorta. And this further bifurcates into branches known as the aortic arches, and this is the developing pharynx. These are the aortic arches from the cranial end of the developing primitive heart tube. So these arches, they branch out and then they wind round the developing pharynx and join with the dorsal aorta on either sides. And each aortic arch represents the arch of that pharyngeal arches or the branchial arches. So the caudal end of the primitive heart is first embedded within the mesenchyme of the septum transversum. So this is what is the septum transversum, the diagrammatic figure you can see. And the caudal end, this one is the sinus venosus. So this is the sinus venosus end. So which gets embedded into the septum transversum and has two horns, the right and left horns. And it receives the blood from the vitelline veins of the yolk sac and the umbilical veins from the placenta and the common cardinal veins from the body wall. So we're going to see about all these things in detail when we talk about the sinus venosus formation. So coming back again to the myoepicardial mantle, which is formed in front, that is ventral to the pericardiac sac, forms the myocardium, and this myocardium, the developing myocardium, will replace the cardiac jelly also and covers the developing endothelial heart tubes. So the endothelial heart tubes itself, so the endothelial tubes persist as the endocardium of the heart, and the myoepicardial mantle will contribute for the myocardium, and the epicardial organ, what we have seen in the dorsal mesocardium, which was there, which covers the myocardium, will be giving rise to the epicardium and the coronary vessels, which supply blood to the heart. And the primitive heart, as we said, starts its beating at day 22, and on day 24 the blood starts circulating throughout the embryo. So this is what is very important for you to remember. And initially, the longitudinal tube, the longitudinal heart tube, the primitive heart tube, presents 4 dilatations. This is very diagrammatic representation for you to know. So the first dilatation is bulbus cordis, next is primitive ventricle, then is primitive atrium, and then is sinus venosus. As we said, the primitive heart tube has 2 circulations first seen, which are the bulbus cordis and the primitive ventricle, which become conspicuous by forming the first constriction. Subsequently, this bulbus cordis will be divided into 3 parts. The proximal part is called the trabeculated part, which is the inflow tract of the right ventricle. Then the conus cordis is the middle 1/3 portion, which is the outflow tracts of both the ventricles, and more distally is the truncus arteriosus, that is the distal 1/3 of the bulbus cordis is the truncus arteriosus, which will develop into ascending aorta and the pulmonary trunk. The distal end of the truncus arteriosus, it will bifurcate to right and left limbs and are connected to the dorsal aorta flanking round the developing pharynx by developing 6 aortic arches. Thus, virtually, the primitive heart tube exhibits 5 saccular dilatations with its constrictions in between. So, the whole of these three put together is the bulbous cordis, and next is the primitive ventricle. So the constriction that appears between the bulbous cordis and the primitive ventricle is the bulboventricular sulcus, which is the first constriction to be appeared. And then there is a constriction between the primitive ventricle and the primitive atrium, which is the atrioventricular canal, where the ventricle and the atrium are in connection with. And the atrium and the sinus venosus are connected by the sinoatrial orifice. So there's a constriction again between the primitive atrium and the sinus venosus, and these two are connected by the sinoatrial orifice. As said, this sinus venosus has two horns, the right and the left, and each horn receives two systemic and one somatic veins. The somatic is the common cardinal vein draining the blood from the body wall, and the umbilical vein, it drains blood from the placenta, and the vitelline veins, they drain blood from the yolk cell. Of these veins, there are few, most of the veins are going to degenerate, leaving behind few veins which form a main part in the fetal circulation. We will see that in general. So here again, coming back, this is the primary heart field and the gray thing is the secondary heart field. So the primary heart field is responsible for formation of the ventricle, atrium and the sinus venosus, and the secondary heart field is mainly for the outflow tract, that is the bulbous cordis, which is influenced by the neural crest cells. And also there are few genes which are very, very important in contributing for the formation of these different aspects in the primitive heart tube. The gene known as HAND1 is important in truncus conus and the left ventricle formation. HAND2 genes are important in the right ventricle. So the cells that are destined to form the right ventricle and left ventricle are very, very different. And TBX5 transcription factor is responsible for the differentiation of the atria. And also the retinoic acid is influencing the venous and the inflow tracts of the developing heart. So this is what is another diagrammatic representation, where it is the truncus arteriosus, then followed by conus cordis, which is not represented here, the bulbous cordis, followed by the primitive ventricle, the primitive atrium and the sinus venosus, where it receives all these veins through the septum transversum. So this is again the differentiation of the heart tube in the same way, in a more clear way rather. So let us see again, because that can be asked to you in viva during the embryology section of your viva. What is being formed from what? So this bulbous cordis, which is the distal most dilatation or the cranial most dilatation, which will be divided further into three parts. The proximal part is the trabeculated part, which forms the primitive right ventricle. So the primitive right ventricle is contributed by the proximal part, that is the proximal 1/3 of the bulbous cordis only, and the middle 1/3 is the conus cordis, which will form the outflow tracts of the right and left ventricles, that is the infundibulum of the right ventricle and the left. And then the distal 1/3 of the bulbus cordis is called as truncus arteriosus, which will give rise to the formation of ascending aorta and the pulmonary trunk. The second dilatation or the circulation is the primitive ventricle. So this bulbus cordis and the primitive ventricle is separated by bulboventricular sulcus, which is the first constriction to be appeared. This primitive ventricle, it forms totally the primitive left ventricle. This will give us the information where in gross, when you see there is hopefully I can draw, when you see the left ventricle is this thick, and when you come on to see the right ventricle, it is a little thinner than the left ventricle, and you can see this thinner, the left ventricle is bulging into the right ventricle. That is because of the fact that the proximal 1/3 is the only part that is contributed for the formation of the right ventricle and is thinner, and the primitive ventricle, the whole will give rise to the formation of the left ventricle. So that's how the myometrium, the myocardium in these right and left ventricles differ. This is the gross diagram what you have seen during the development, the blood supply of the heart. And then comes the constriction, the primitive atrium, where the ventricle and atrium are in continuation with the atrioventricular canal, and this primitive atrium will be giving rise to the right and left atrium. And the blue one is the sinus venosus, which has a body and two horns, and this body of the sinus venosus will be absorbed into the right side of the primitive atrium to form in the development, to contribute in the development of the right atrium, the smooth part of the right atrium. And the right horn and the left horn will receive all these veins which we were talking about, the two vitelline veins from the yolk sac, two umbilical veins from the placenta, and the two common cardinal veins from the body wall. So the anatomical segmentation of the heart tube, as said, is started from day 22. So the looping starts from day 23, because the looping starts because there is inclusion of new cardiac cells from the secondary heart field. So the heart tube has the following regions. This is the cranial end, which is the outflow, that is the arterial end, and the caudal end, it is the inflow, which is the venous end. And this is the bulbus cordis, followed by the ventricle and the atrium and the sinus venosus. On day 22, if you see, the heart tube which is within the pericardial cavity is only the bulbus cordis and the primitive ventricle, which are the primary circulations with the first constriction, that is the bulboventricular sulcus, that has been formed, are present within the pericardial cavity. So the atrium and the sinus venosus, the other parts of the heart, also should be included into the pericardial cavity, where the heart tube now has to undergo the looping, so that whole of the heart tube is seen within the primitive pericardial cavity. So this cardiac loop formation is necessary because the outflow region, it lengthens more. The bulbus cordis there is lengthening of the bulbous cordis part of the developing heart more because the cells are added from the secondary heart field. That is because the secondary heart field is concerned mainly with the outflow tracts. And as these lengthen and to accommodate the growing primitive heart tube in its length, the looping has to occur. So the ends of the tube are fixed in place, causing the tube to loop because the ends can't extend beyond its level because the cranial end is stationed there because of the connection of the arterial arches back to the dorsal aorta, and the venous end is restricted in its further growth, increasing in its length by the septum transversum that is present caudal to the sinus venosus, through which all these veins, the common cardinal vein or the duct of Cuvier and the vital line and the umbilical veins are drained into the sinus venosus. So whatever has to happen should happen within the pericardial cavity, the lengthening. So to accommodate the growing heart, the looping and the bending of the heart tube is mandatory like this to accommodate into the pericardial cavity. So along with this lengthening, there are series of constrictions and expansions that are formed in the heart tube. What we have seen now, from cranial to caudal, it is the bulbous cordis, which in turn is divided into three parts, then the primitive ventricle, then the primitive atrium, and then the sinus venosus, which are all interconnected by various orifices. And these expansions, they contribute for the different chambers of the heart. And after the looping and bending that happens, so the bulbous, the outflow tract, the bulbous cordis is seen here, the primitive ventricle and the primitive atrium, and this has to take its normal anatomical positions after the looping that occurs. So as said earlier that the heart beats by day 22 and the circulation starts by day 24, which is the prime requisite for the developing primary heart tube also. So initially the contractions of the heart are myogenic. So the muscle layers of the atrium and ventricle are continuous, and the contractions occur in peristaltic wave starting from the sinus venosus, atrium, ventricle, bulbous cordis, so on so forth. That's how the blood flows. So there's an ebb and flow type of circulation that is seen in the primordial heart tube, where the blood enters through the sinus venosus, atrium, ventricle, bulbous cordis, and out of the outflow tract. And so the blood enters the sinus venosus from the embryo through the common cardinal veins or the duct of Cuvier. That means the common cardinal veins are also known as duct of Cuvier. Developing placenta through the umbilical veins, and from the umbilical vesicle, that is the yolk sac, through the vitelline veins. So blood from the sinus venosus enters the primitive atrium or the primordial atrium. Flow from it, it's controlled by sinoatrial orifice, which develops the valves, that is the sinoatrial valves. The blood then flows through the atrioventricular canal into the ventricle, and from there the ventricle contracts and the blood is pumped through the bulbous cordis, into the truncus arteriosus, and then into the aortic sac, from where through the aortic arches, which are the arches of each pharyngeal arch, are then passed on to the systemic circulation. So as the development progresses, the longitudinally placed saccular heart tube, which one sacculated heart tube, now grows rapidly under the influence of the neural crest cells, which influence the secondary heart field and which gives more cells to the outflow tract, that is more so for the bulbous cordis. So bulbous cordis mainly grows in length, and eventually the tubular heart bulges ventrally and somewhat caudally into the pericardial sac. So this sacculated heart, once there is enormous growth in the bulbous cordis region and has to accommodate in this area, the heart first grows ventrally and caudally and indents into the pericardial sac in a U-shaped manner. So this cardiac loop is mainly formed by the bulbous cordis and the ventricle. So this loop is called as bulboventricular loop. So this is the bulbous cordis part here, and this is the primitive ventricle. This is the primitive atria and the sinus venosus. So loop mainly is formed by bulbous cordis and the primitive ventricle. So this loop is called as bulboventricular loop. The cephalic end of the loop is formed by the bulbous cordis and its subdivisions and is displaced caudally, ventrally and to the right. The caudal limb is formed by the primitive ventricle, this one. The caudal limb is formed by the primitive ventricle, which is displaced to the left, while the primitive atrium, which is present here, will be shifted posterior-superior to the primitive ventricle. This loop formation will be completed by the end of 4th week, and the shifting of the atrium posterior-dorsally or posteriorly to the primitive ventricle, when it gets shifted onto the left, is facilitated by formation of the transverse sinus, that is degeneration of this dorsal mesocardium, where the pericardial cavity is present all around the developing heart tube, facilitates the looping of the caudal part of the primitive heart tube. So why looping is again to accommodate the developed heart into the pericardial cavity. So once there is addition of cells to the bulbous cordis region by the secondary heart field under the influence of neural crest cells, the bulbous cordis is bent onto the right side ventrally and caudally like this, and that's how the bulboventricular loop is formed. Till this bulboventricular loop is formed, the atria and sinus venosus are outside the pericardial sac because the transverse sinus has not yet formed. So once the transverse sinus has been formed and the dorsal mesocardium regresses, then there is space for the atrium to enter into the pericardial cavity. So once this bulbous cordis moves ventrally downwards onto the right and the primitive ventricle moves onto the left, the primitive atrium will move dorsally and cranially to the primitive ventricle, entering into the pericardial sac. So this bulboventricular sulcus is an external demarcation between the bulbus cordis with the primitive ventricle.